Picture this. It is 2008. A team of researchers is excavating a cave in the Altai Mountains of Siberia, a place called Denisova Cave, named after an 18th century hermit who used to live there, which is either charming or slightly unsettling depending on how you feel about hermits. The cave has been occupied on and off for hundreds of thousands of years by various human species, which makes it an extraordinary archaeological site. The team is working carefully, methodically, doing what paleontological fieldwork looks like when it isn't being dramatized for television. Mostly sitting in the dirt, brushing things very slowly with small brushes, and trying not to sneeze on anything. And then someone finds a bone fragment. Not a skull, not a jaw, not even a complete bone, a fragment of a finger bone. The distal phalanx of a fifth digit, which is the scientific way of saying the tip of a little finger, from an individual who appears to have been somewhere between 5 and 13 years old at death. The fragment is approximately 3 centimeters long. It is, by the measure of paleontological significance, a completely unimpressive piece of material. Except it isn't. Because when that finger bone fragment is sent to a laboratory in Leipzig, Germany, and a team of geneticists extracts and sequences the ancient DNA preserved inside it, what they find does not match any known human species. It is not Homo sapiens. It is not Neanderthal. It is something else entirely, a previously unknown branch of the human family tree that had been sitting completely undetected in the fossil record, invisible to generations of paleoanthropologists, right up until the moment that molecular biology walked into the cave and introduced itself. The Denisovans had arrived, and they were unlike anything anyone expected. Before examining what makes the Denisovans so extraordinary, establish the context of what was known about human prehistory before their discovery, because the magnitude of the surprise requires that backdrop. By the mid-2000s, the hominin fossil record had been studied intensively for over a century. Dozens of species had been named, described, debated, and placed in various proposed relationships within the human family tree. The broad outlines of human evolution, African origins, successive waves of dispersal, the relationship between archaic and modern humans, had been argued through mountains of fossil evidence, stone tool assemblages, and the early results of ancient DNA research. Neanderthals, specifically, had been genomically characterized enough to establish their close but distinct relationship to modern Homo sapiens. The field felt, if not complete, at least comprehensively mapped in its major features. The Denisovan discovery demonstrated that this confidence was premature. A human lineage that had apparently occupied a significant portion of Asia, from Siberia to the Tibetan Plateau to Southeast Asia. For hundreds of thousands of years had left almost no identifiable physical remains in the fossil record. While paleoanthropologists were meticulously cataloging every jaw fragment and cranial shard from European and African sites, an entire human species was hiding in the Asian fossil record, its distinctive identity invisible, because the morphological differences from known species were insufficient to flag the remains as something requiring special explanation. The detection required ancient DNA. Without molecular analysis, the Denisovan remains would have been classified as unremarkable, possibly attributed to late archaic Homo sapiens or ambiguous archaic humans of uncertain affinity. The genome was the discovery. The physical bones were almost incidental to the revelation. The genetic picture of the Denisovans that emerged from subsequent analysis is simultaneously detailed and maddeningly incomplete, which is the paradox at the heart of what makes them so mysterious. The Denisovan genome was sequenced to high coverage from the original finger bone and subsequently from additional specimens. A tooth, another tooth, a partial jawbone found in Tibet, making their genetic profile one of the better characterized archaic human genomes in the research literature. From this genomic data, a great deal can be inferred. Their population history, their relationship to Neanderthals and modern humans, the timing of their divergence from other hominin lineages, and most remarkably, their presence as a genetic component in living human populations today. Denisovans diverged from the lineage leading to Neanderthals approximately 400,000 years ago, after that combined lineage had already diverged from the ancestors of modern Homo sapiens. They are therefore more closely related to Neanderthals than either is to modern humans, but they are sufficiently distinct from Neanderthals that their genomes cluster separately in any phylogenetic analysis. The population that eventually became Denisovans appears to have originated in Africa and dispersed into Asia, where they remained as a separate lineage for hundreds of thousands of years, while Neanderthals occupied Europe and Western Asia. The Denisovan genome reveals genetic diversity suggesting that the Denisovan population was not a small isolated group, but maintained a population large enough to accumulate substantial within-group genetic variation over their existence. There appear to have been multiple distinct Denisovan populations. Analysis of different specimens suggests that the Siberian Denisovans and the Tibetan Denisovan and the populations that ultimately contributed DNA to living Southeast Asian and Pacific Island humans may have been as genetically distinct from each other as different subspecies of modern humans. The Denisovans of the fossil record may have been a diverse group of populations spanning enormous geographic and temporal ranges, loosely grouped under one name because that one name is all the physical evidence currently supports. The interbreeding story is where the Denisovan discovery shifts from fascinating to transformative in its implications for understanding human prehistory. Modern humans carry Denisovan DNA. This is not a subtle statistical signal requiring careful mathematical extraction. In some populations, it is a substantial component of the genome. Aboriginal Australians and Melanesian populations carry approximately 4 to 6% Denisovan ancestry, making them among the populations most strongly shaped by interbreeding with Denisovans. Some populations in the Philippines carry even higher percentages. East Asians carry a smaller but detectable Denisovan contribution. The Denisovan genetic legacy is distributed across a wide swath of the Pacific region in a pattern that implies multiple interbreeding events between expanding Homo sapiens populations and Denisovan groups they encountered during the dispersal of modern humans through Asia and into the Pacific. The functional consequences of this interbreeding are not trivial. The most dramatic identified example is the EPAS 1 gene variant found at high frequency in Tibetan populations, a genetic adaptation to high-altitude hypoxia that allows Tibetans to maintain normal hemoglobin levels at altitudes that cause serious physiological problems in other humans. This variant, which is functionally important and strongly selected in the Tibetan population, was inherited from Denisovans. The Denisovans, who occupied the Tibetan plateau, as evidenced by the Siahe mandible found there at an altitude of 3,200 meters, had apparently spent enough generations at high altitude to evolve local adaptation to hypoxia, and modern humans moving into Tibet inherited that adaptation through interbreeding rather than having to independently evolve it. This is adaptive introgression, the transfer of beneficial genetic variants between species through interbreeding, and it represents a mechanism by which modern humans effectively borrowed evolutionary solutions from other hominin species they encountered during their expansion. The Denisovan altitude adaptation in Tibetans is the clearest example, but research has identified other Denisovan-derived variants in immune function genes, metabolic genes, and genes related to cold adaptation in populations carrying Denisovan ancestry. The Denisovans did not simply disappear. Their genetic contributions are functioning in living human bodies right now. The physical appearance of Denisovans is the dimension of their story where the mystery becomes most acute, because the genome can tell you many things, but reconstructing the face and body of a species from its DNA requires methods that are only beginning to approach the question with any rigor. A 2019 study attempted to predict Denisovan skeletal anatomy from patterns of gene regulation in the Denisovan genome, using what's called a methylation map of the Denisovan genome to identify genes where Denisovan regulatory patterns differed from those of modern humans and Neanderthals, then inferring what anatomical differences those regulatory differences would produce. The results painted a picture of a large-bodied hominin with broad features, possibly a wide skull with a low forehead and a robust facial structure. The reconstruction suggested Denisovans might have looked somewhat similar to Neanderthals in general body plan, but with specific differences that would have been visually apparent. But this reconstruction is deeply uncertain. The methodology depends on assumptions about how gene regulation maps to anatomy that haven't been fully validated, and the prediction confidence diminishes for features where the regulatory differences are subtle or where the gene-phenotype relationship is complex. The physical description of Denisovans remains more constrained by what the genome implies than confirmed by direct morphological evidence. The physical fossils themselves, a small handful of specimens from two sites, are tantalizing precisely because they include so little. By the way, if you want to go even deeper into the secret lives of ancient humans, I just launched a full course called The Secret Lives of Ancient Humans. 10 modules, 3+ hours of content covering everything we never talk about on YouTube. What they feared, how they communicated, what hunted them, and much more. Plus an exclusive PDF guide included. Link in the description. See you inside. The finger bone is morphologically consistent with either a female modern human or a female Neanderthal. The teeth are large, larger than Neanderthal teeth in most measurements and much larger than modern human teeth, suggesting either a generally large body size or at least a robust dentition. The Xiahe mandible from Tibet is large and robust, lacking a chin, broadly consistent with archaic human morphology, but not specifically distinguishable from other archaic humans on morphological grounds alone. This is the fundamental problem. If you handed a Denisovan fossil to a paleoanthropologist without telling them what it was, and without the possibility of DNA extraction, there is a real possibility they would classify it as a regional variant of archaic Homo sapiens, or an unusual Neanderthal, or a representative of one of the ambiguously classified Asian archaic human fossils that have accumulated in the literature over decades. The Denisovans may be hiding in plain sight in museum collections, their identity obscured by the limitations of morphological classification. That possibility, Denisovan fossils already sitting in collections, misclassified, is one of the most active areas of current research in paleoanthropology, and has produced some specific candidates that have dramatically expanded the geographic and temporal range of what Denisovans might represent. Several enigmatic Asian fossil specimens that resisted confident classification for decades have been proposed as potential Denisovans or Denisovan-related populations, on the basis of morphological features consistent with the genomic predictions and geographic and temporal contexts consistent with known Denisovan presence. The Harbin cranium, sometimes called Dragon Man, found in northeastern China and dated to possibly over 100,000 years ago, is one proposed candidate. It is extraordinarily large, morphologically archaic in multiple features, and doesn't fit comfortably into any established hominin species as currently understood. Analysis has placed it within or close to the Denisovan branch of the human family tree, though this assignment remains contested. The Dali skull from China, the Jinyushan skeleton, and various other fragmentary Asian specimens from the Middle to Late Pleistocene have all been discussed as potential members of an expanded Denisovan group or closely related populations. The consistent pattern across these specimens is size. They tend to be large, robust, with features intermediate between earlier archaic humans and the distinct characters of Neanderthals or modern humans, and geographic distribution across Eastern and Southeastern Asia in exactly the range where Denisovan ancestry is strongest in living populations. If any of these identifications are confirmed through ancient DNA recovery, which is technically challenging from the warm, humid environments of most of Southeastern Asia, where DNA preservation is substantially worse than in the cold, dry conditions of Siberia, the Denisovan fossil record would be transformed from a handful of tiny fragments to a substantial collection of specimens spanning hundreds of thousands of years of Asian prehistory. The species would shift from the most physically cryptic in the human family to one with a detailed morphological record. The mystery would not be solved. It would deepen because a detailed physical record would raise new questions about diversity, range, and regional variation, but its character would change dramatically. The geographic distribution inferred from genetic evidence in living populations is one of the most surprising aspects of Denisovan prehistory, and implies a degree of ecological flexibility that challenges assumptions about archaic hominin adaptive range. Denisovan ancestry is highest in populations from Australia, New Guinea, and the Pacific Islands. Populations whose ancestors arrived in those regions during the initial dispersal of modern humans through Southeast Asia approximately 50 to 60,000 years ago. For these populations to carry high Denisovan ancestry, interbreeding must have occurred somewhere along the dispersal route, in a geographic zone consistent with the pathway from the Middle East through South Asia and into Southeast Asia and the Pacific. This implies Denisovan presence in Southeast Asia, in tropical rainforest environments dramatically different from the cold steppe environments of the Altai Mountains where physical Denisovan remains have been found. The same species or related populations that occupied Siberian cave sites and the Tibetan Plateau were also present in the equatorial forests of Southeast Asia, if the genetic evidence implies what it appears to imply. This is an extraordinary biogeographic range, cold temperate to alpine to tropical, suggesting either a highly adaptable species capable of thriving in fundamentally different ecological conditions, or a diverse set of populations that had diverged sufficiently to occupy different niches while remaining close enough genetically to interbreed with modern humans when they arrived. The ecological flexibility implied by this range is, if confirmed, remarkable. Most hominin species in the fossil record appear to have been more ecologically constrained. Neanderthals were primarily cold-adapted and concentrated in Europe and Western Asia. Earlier, Homo species tended to cluster in specific environmental zones. A Denisovan presence from Siberia to the tropics would make them among the most ecologically generalist archaic humans known, which adds another dimension of strangeness to their near-total absence from the physical fossil record. The Denisovans' ultimate fate, why they disappeared as a distinct lineage, is a question the genomic record addresses partially, but not conclusively. Modern humans expanding through Asia encountered Denisovan populations and interbred with them at multiple points. The timing and number of these interbreeding events can be partially reconstructed from the pattern of Denisovan ancestry in contemporary populations. The high Denisovan ancestry in oceanic populations suggests one major interbreeding event during the dispersal of modern humans into Southeast Asia and the Pacific. The lower Denisovan ancestry in mainland East Asian populations suggests either less intensive contact, later contact after some dilution had occurred, or contact with different Denisovan populations that were genetically less similar to the ancestors of oceanic populations. Whether Denisovans disappeared through competition with incoming modern humans, through absorption via interbreeding, through environmental change, or through some combination of these factors cannot be determined from current evidence. The pattern of their genetic legacy in living populations is consistent with gradual demographic dilution. Modern human population sizes expanding while Denisovan populations remained smaller, resulting in the Denisovan genetic contribution being progressively reduced to a minority component over generations. But this is reconstruction through statistical inference, not direct observation. What can be said is that the process was not a clean extinction. Denisovans did not simply vanish and leave modern humans unchanged. They merged, partially unevenly, in different proportions in different geographic regions, with the modern human populations that encountered them. Their genes are functioning in living people. Their adaptive contributions to modern human biology are demonstrable. Their disappearance as a morphologically and genetically distinct lineage is real, but they left a biological legacy that has persisted for 60,000 years and continues to influence the health, physiology, and adaptation of living human populations today. The broader lesson that Denisovans have taught paleoanthropology is perhaps more important than any specific finding about the species itself. The human family tree is vastly more complex and populous than the physical fossil record suggested. And molecular biology has provided a window into that complexity that morphology alone cannot open. Before the Denisovan discovery, the assumption that a human lineage occupying a significant portion of Asia for hundreds of thousands of years would be detectable in the fossil record seemed reasonable. After the Denisovan discovery, that assumption is no longer defensible. An entire human species was effectively invisible to a century of paleoanthropological investigation, detectable only through the genetic legacy it left in modern human genomes, and through the genomic analysis of specimens too small to have attracted special attention through morphological examination alone. This implies that other undetected hominin lineages may exist, species or populations that contributed genetic material to modern humans or to each other, left small or unrecognizable physical traces, and await discovery through the ongoing expansion of ancient DNA research into broader geographic and temporal ranges. The ghost lineages inferred from unusual patterns in modern human genomes, population genetic signals implying ancestry from unsampled archaic groups, suggest that the Denisovans may not be the last such discovery. A potential ghost lineage has been inferred in West African modern human populations, where genome analysis has identified a component of ancestry that doesn't match any known archaic human group. Not Neanderthals, not Denisovans, but some other archaic lineage that interbred with the ancestors of some West African populations before those populations were part of the modern human diaspora. No physical remains attributable to this inferred lineage have been identified. It may await a fingerbone fragment in the right cave, the right sediment, the right preservation conditions, discovered by the right researcher with the right molecular biology laboratory, waiting to receive it. The Denisovan story is, at its core, a story about the limits of what we thought we knew and the tools that expanded those limits. For most of the 20th century, the story of human evolution was written in bone and stone, the physical remnants that survived geological time well enough to be found and studied. That story was not wrong, but it was incomplete in ways that the physical evidence couldn't reveal from within its own methods. The addition of ancient genomics to the paleoanthropological toolkit has done something extraordinary. It has made the invisible visible. Lineages that left no morphologically distinctive physical remains. Populations that were absorbed rather than replaced. Interbreeding events that left no archaeological signature but permanently altered the genetic makeup of the descendants of the participants. All of these are now accessible to investigation in ways they simply weren't before. The little fingerbone from Denisova Cave was 3 centimeters long. The 5 to 13-year-old child it belonged to lived and died in a Siberian mountain cave approximately 50 to 80,000 years ago, completely unaware that their existence would one day be unknown, then detected, then investigated with tools that would have been incomprehensible magic to every human alive at the time. The child left essentially nothing. No art, no identifiable tools, no skeletal features that would have attracted special attention, no unambiguous cultural signature. Just a fingerbone with DNA inside it. And inside that DNA, an entire species that the human family had forgotten, preserved in molecular form, waiting for the moment when our descendants would be sophisticated enough to read it. That moment arrived in 2010, when the first Denisovan genome was published. The most mysterious human species had finally introduced itself, 60,000 years after its last members died, or merged into the populations of the people who came after. They had been there the whole time. We just didn't have the right questions yet.